Good afternoon, dear colleagues. Welcome to this ERKNet webinar. My name is Elena Levchenko, and I am speaking from my office in Amsterdam. Before starting, let me remind you that your microphones are turned off, but during and after the webinar, you can send your questions through the GoToWebinar application that you see on your screens. I will ask your questions at the end of the presentation, and we'll try to answer them all. The webinar will last approximately 30 minutes, and then we will have 15 minutes available for questions. So I'm happy to announce our speaker of today, Dr. Guillaume Dorval. Dr. Dorval is trained as a pediatric nephrologist in the Division of Pediatric Nephrology in the Hôpital Necker Enfants Malades in Paris, but also as a genetician in the lab of Professor Corinne Antignac. And he is now working as an assistant professor at the University of Paris in genetics, and also as a physician in the hospital Hôpital Necker Enfants Malades in Paris as well. So he has a double specialty, and he is really a specialist in the topic of today, which is Galloway Mowat syndrome. As you know, the first genetic defects in this syndrome were discovered by the group of Corinne Antignac. And so Guillaume will talk about this very interesting disease. So Guillaume, welcome. We are eager to hear your talk, please.
Thank you, Elena. Good afternoon, everybody. It's a real pleasure to be here this afternoon just to talk about the Galloway Mowat syndrome, and the lab in which I work is really involved in the Galloway Mowat syndrome and in all the study about the pathogenesis of Galloway Mowat syndrome and diagnosis, genetic diagnosis of Galloway Mowat syndrome, and especially Corinne Antignac and Géraldine Mollet, who both did a huge work about this disease. So as you all know, the genetic studies of hereditary nephrotic syndrome has led to the identification of more than 50 genes that, if mutated, are responsible for monogenic forms of tRNA, either isolated or syndromic. And most of these genes encode proteins expressed in the podocyte with various functions, such as transcription factors, mitochondrial proteins, enzymes, but particularly structural proteins of the slit diaphragm, as well as actin binding regulator. And mutation in these genes can lead to either an isolated or a syndromic nephrotic syndrome, or even non-nephrotic range proteinuria. And the organ specificity may be due to a tissue-specific gene expression, leading to a phenotype in certain tissue, but not in other tissues. But it can also be due to the involvement of specific pathway in some specific tissues. So at this step, I think it's really important to note that we can find some similarities in the architecture and physiology of some neurons and the podocytes, which are both terminally differentiated cells and very specialized cells. And these similarities can explain the specific phenotypes involving in neuro and renal disorders. So probably due to these trend similarities, some syndrome associate tRNA and neurologic involvement, and including both central or peripheral neurologic involvement. And you have a lot of different syndromes, but today I will focus on the Galloway Mowat syndrome that has been linked to different genes, and we will focus on these different genes that lead to GMS. So the GAMOS, the GMS, is an extremely rare genetic disorder, and since its first description in 1968 by Galloway and Mowat, approximately 100 cases have been published. And this autosomal recessive disorder is characterized mostly by sRNA, microcephaly, and central nervous system abnormalities. And we can find a very huge clinical heterogeneity in this syndrome, with microcephaly that can be primary when detected at birth or secondary when it develops postnatally. The renal disease ranges from isolated proteinuria to nephrotic syndrome that processes to end-stage kidney disease with a variable onset, like congenital nephrotic syndrome or later during childhood. And renal lesion may be DMS, so diffuse mesangial sclerosis, or FSGS. Neurological manifestation and brain abnormalities are various and include developmental delay, hypotonia associated to cortical and or cerebellar atrophy, and gyration and myelination defects. And most affected children do not survive beyond teenage years, with the commonest causes of death being nephrotic syndrome and seizures. So to date, we have 11 genes that have been identified to be mutated in GMOS. And the most frequent mutated genes are OSGEP in the KIOPS complex and WDR73, and we'll focus on these different genes in the presentations. And so most genes encode proteins involved in various processes of RNA metabolism, but can also encode proteins involved in nucleocytoplasmic transport or in transcriptomal regulation, such as the last gene described, that is PRDM15. And as you can see, we have a few families for each gene. So it's really a rare disease focusing on OSGEP and WDR73. And interestingly, the type of mutations can give us some insight into the critical role of the altered protein. And for example, we can't find any biallelic loss of function mutations in the OSGEP gene. And that means that biallelic loss of function mutations are probably lethal for patients. And a particular fact is that whereas mutation in KIOPS related genes or WDR73 are only found in GMOS, in some cases mutations in other GMOS genes could also be found in isolated sRNAs, for example in NUP107 or NUP133, or isolated neurological involvement, or even in other diseases such premature ovarian failure and acute actually, it's very difficult to understand this heterogeneity depending on the gene and the genetic variation. So let's start now with the genetic basis of the GMOS, and starting with the first gene that has been described, that is the WDR73 genes. It has been described in a particular subset of GMOS affected children. And indeed, patients present mainly with postnatal progressive microcephaly, ataxia with cerebellar atrophy on the MRI, and sometimes epilepsy and optic atrophy. Regarding the kidney, the renal disease in these patients tend to be later and more variable age of onset compared to patients with mutation in other GMOS-associated genes, and is absent in some young adult patients. So very variable clinical findings, notably regarding the kidney involvement. And this protein is a protein composed with WD domains, and WD domains are known to be involved in protein-protein interactions. And so while WDR73 was the first gene described in GMOS, it's also the less characterized gene with different putative roles in disease development. And the protein localized in the cytoplasm during the interphase, and during mitosis, the protein localized to the spindle poles and astral microtubules, like here, that suggest a role in the cell cycle. And initial studies have shown a role of this protein in the cell cycle regulation, in the cell survival, as well as in microtubule dynamics, but the role is still unclear for that. And we had a zebrafish model, and that has been published in 2015. And in this zebrafish model, invalidation of this gene, the WDR73 with morpholino, revealed a significant brain growth and morphology defects, as well as smaller and poorly differentiated midbrain and cerebellum, indicating the crucial role of this gene, the WDR73, in the survival of neuronal progenitors. And to complete the role of the functional role of this gene, a few years ago, Frances Tilley, who was a postdoc in the lab, demonstrated an interaction between this specific gene and this protein, WDR73, sorry, and protein from the integrator complex, and namely protein INS9 and INS11. And both proteins are the catalytic subunits of the integrator complex, that is a large RNA polymerase II binding complex with various roles in RNA metabolism and transcriptional control. And most interestingly, mutations in genes encoding the integrator subunits, INS1 and INS8, have been linked to neurological disorders that is strikingly reminiscent of the affecting GMOS patients with WDR73 mutations. And Frances then showed that the WDR73 gene and the protein is implicated in two integrator-regulated cellular pathways. First, the processing of some specific RNAs that are called the iridylate-rich small nuclear RNAs that are mandatory for the constitution of the spliceosome. And she showed an effect of the protein of the mutation, the WDR73 pathogenic variant, in neural progenitors, but not in podocytes. And she showed that what she showed suggests that we can find different pathways specific to the cell line, and this pathway is not involved in the podocyte. And then as the integrator complex is known to regulate the transcriptional response of to the EGF stimulation, she showed a defect in the response of transcription for example, the FOS gene, that is a gene known to be sensitive to EGF, with an even more important decrease of response here with WDR73 than with the integrator 11, the ENTS 11. And it suggests that through the integrator complex, the gene WDR73 and the protein may act as a very important regulator of some transcriptional pathway. And so she moved to the transcriptomic level just to try to decipher the pathogenesis of this gene. And she showed that in cells invalidated for the WDR73 or the protein from the integrator complex, the ENTS 11, only specific genes were dysregulated. And specifically, the upregulation of the gene encoding the cyclin D1, as shown here by the RT-PCR, as well as the other G1 phase cyclins, for example, the cyclin D2 and the cyclin D3. And she showed the downregulation of the gene encoding the P21, and the P21 is a cyclin D1 inhibitor. And so altogether, this result suggests a role for the WDR73 gene in maintaining the neuron and the podocyte differentiation state by inhibiting the re-entry into the cell cycle to maintain the differentiate state. So let's move now to the KIOPS complex. The KIOPS complex that have been published 3 years later, so in 2017. And this work was a collaboration with the group of Frida Hildebrandt, and the group identified 22 mutations in different genes. And the genes are LAGE3, the OSGEP gene, the TP53 ERKA gene, and the TPRKB gene that encode the 4 subunits of the multi-protein complex KIOPS. And these mutations were found in 37 individuals with Galloway Mowat syndrome from 33 unrelated families. And the majority of the mutations were identified in the OSGEP gene, as I said just before, and we never found biallelic loss of function mutation. So remind that it really suggests that the total absence of this protein, and in particular the OSGEP protein, is not compatible with life. So all the patients with KIOPS mutations presented with a very severe phenotype, with death occurring at a median age of 6 months. And patients had primary microcephaly with developmental delay, hypotonia, and seizures. And MRI abnormalities were mainly cortical and cerebellar atrophy. As you can see here, the cortical and the cerebellar atrophy. And duration defects here, such as polymicrogyria and myelination defects. And the renal disease was also very severe, with early onset proteinuria at median age of 3 months that rapidly progressed to end-stage kidney disease at a mean age of 11 months, so before 1 year. And histological lesions found were mainly the FSGS lesion and the diffuse mesangial sclerosis in patients with, of course, food process effacement in electron microscopy. So the mutations in the KIOPS genes are really linked to a very severe phenotype. What is the KIOPS complex? The KIOPS stands for the kinase endopeptidase and other protein of small size. And so all the protein encoded are the LAGE3 and TPA-kappa B, that are the protein of small size, and the kinase TP53RK and the OSGEP protein. And there is a fifth subunit that is called Gun7 that was only known to be present in yeast, but there was no ortholog in human at this stage. And the KIOPS complex is involved in multiple cellular processes essential for sustained life, and it plays a role in telomere maintenance and transcriptional regulation. And later, another function that was not aware of before this work was assigned to KIOPS. It was the biogenesis of the T6A modification on the tRNA, and we will talk about the tRNA now, because it plays a major role in the pathophysiology of GMOS. So KIOPS is involved in the multiple cellular processes essential to sustain life, at least in yeast. So now, what are the tRNA modifications and their role in human disease? The transfer RNAs, so the tRNAs, are key players of protein synthesis, and they can decode the genetic information organized in the mRNA, so in the messenger RNA codons. And so they can specifically recognize all codons and translate the codons into the code of 20 amino acids. So they are the codons in the mRNA, and they can recognize the codon and translate it into an amino acid. And to be fully active, tRNAs must undergo extensive post-transcriptional modifications that catalyzed by different enzymes, as you can see here, and you can recognize here, for example, at the position 37, that the KIOPS complex is mandatory for post-transcriptional regulation modification here and here, because we'll talk about that later. And at the position 48, I think it's 48. I can't see very well, but the WDR4 that is really involved also in the post-transcriptional modification here. And we know that lack of these modifications decrease the translation fidelity and accuracy, contributing to protein aggregation with deleterious consequence to the cell, and notably ER stress activation. I mean, UPR activation. And the recent work show that the tRNA hypomodification occur in several diseases such as mitochondriopathies and Alzheimer and other metabolic disease. And GMOS is the first example of involvement of such a mechanism in renal disease. So what is exactly the T6A modification? The T6A stands for the N6 threonyl carbamoyl adenosine modification. So it's a barbarian name, but it's one of the few universally conserved tRNA modification across species. So very important tRNA modification, and it adds a threonyl carbamoyl moiety on position 37 of one tRNA, and the adding of this specific threonyl carbamoyl moiety allow to read codons that start with an adenoside, and so it's of fundamental importance. So it includes the most famous codon that you all know, that is the ATG or AUG in the RNA codon that code for the initiator methionine, and so to start all proteins. And so it plays a crucial role in the efficiency and fidelity of the initiation of translation impacting protein biosynthesis and cellular proteostasis. And the biosynthesis of T6A is catalyzed by two universally conserved proteins, the YRDC, that leads to the synthesis of the threonyl carbamoyl AMP. So the first step of this modification, but is not involved in the KIOPS complex, and the KIOPS complex, and particularly the OSGEP protein in the KIOPS complex that will transfer the threonyl carbamoyl to the tRNA. But the role of the KIOPS in human remained unknown. And so Géraldine Mollet from our lab performed a knockdown of KIOPS components, including the OSGEP and the TP53RK and TP-RKB in human podocyte cell line. And she first showed a decrease in proliferation, in migration and survival, and an increase in apoptosis of cells harboring the OSGEP mutants. And she also demonstrated a decrease of the T6A levels and de novo protein synthesis as a reflect of the translation defect. And so she also showed an activation of the unfolded protein response, that is an intracellular pathway involved in case of endoplasmic reticulum stress, and that reflects a translation fidelity defect. And so to assess the pathogenic effect of the OSGEP mutations, she also performed yeast growth complementation studies, and she used yeast depleted for KI1, and KI1 is an ortholog of OSGEP in the yeast, and she identified two classes of OSGEP mutant alleles. She identified the hypomorphic alleles in the green box that restored growth, but to a lesser extent than wild type OSGEP, and she described the amorphic alleles in the red box here that were completely unable to restore growth exactly as the empty plasmid. And she says measured the T6A levels in these yeast, and she was able to correlate the T6A level to the efficiency of rescue in yeast, and the yeast expressing amorphic alleles has a very low level of T6A compared to those expressing the hypomorphic alleles. And using available crystal structures from homologous KIOPS complex, she constructed a 3D model of the human KIOPS and projected the various mutations onto the crystal structure, and it allowed to show that the amorphic mutation were mainly located near to the catalytic site of the OSGEP protein. And so to complete this part on KIOPS, I just want to show that there are several animal models for this kind of mutation that have been generated, such as the zebrafish model, mouse model, and I will show you a Drosophila model. And not surprisingly, OSGEP knockouts display embryonic lethality, whatever the model, and was not viable. We could observe microcephaly that was found in mice and zebrafish models here and here, microcephaly. And in zebrafish, the tunnel staining of brain sections demonstrated an increase in apoptotic cells in the cortex of homozygous fishes compared to the heterozygous one. And in Drosophila, by conditional inactivation of the TCS3 gene, that is the OSGEP ortholog in Drosophila, the group of Daniela Brown recently showed that we can observe slits diaphragm defects, and they confirmed the importance of ER stress, and especially the involvement of the transcription factor ATF4 in the development of podocyte lesions. And so they did all this work in nephrocytes, Drosophila nephrocytes, and they showed that inhibition of the response to ER stress by inhibition of PERK or ATF4 led to a partial rescue, as you can see here and here, a partial rescue of the nephrocyte injury and the slit diaphragm defect. So it really confirmed the real importance of the ER stress in the pathogenesis of the podocyte injury in this disease. And interestingly, there is one particular variant that is associated with a neurological defect and quite similar to other variants, but a different renal phenotype, including hypomagnesemia, a non-nephrotic proteinuria, but without renal insufficiency. And so we can't exclude the fact that we could find some, at least OSGEP variants in some other renal phenotypes. And now we have to talk about the Gun7. Gun7 is the fifth element known in yeasts that I told you just before, but that was never described in humans. And so the question was, are Gun7 mutation the cause of GMOS? And some of you may know that Corinne Antoniak had identified more than 10 years ago a mutation in a gene with a barbarian name called C14ORF142 or 142, and she identified this mutation in this gene in 4 consanguineous families from the Ores region in Algeria, with exactly the same stop mutation at the very beginning of the gene and of the protein. And patients presented a more attenuated form of the disease, but GMOS phenotype compared to those mutated for the KIOPS genes, with secondary microcephaly associated to developmental delay, hypotonia, and with major MRI findings that were cortical and cerebellar atrophy and hypoplasia, a thin corpus callosum, ventricular dilation, and some patient and myelination defects. But we never found any duration defects. And regarding the kidney, the onset of proteinuria was between 2 and 5 years, and the end-stage kidney disease was reached between 2.5 and 6 years. And we could find histological lesion and kidney biopsy that range from DMS to FSGS, exactly the same as we find in other KIOPS genes. And at that time, we are very disappointed because the encoded protein was really unknown. And so it was not possible to do a link between the KIOPS and the KIOPS complex and this protein. And this protein is a very small protein. And so antibodies didn't work for functional studies, and the knockout mouse model had no phenotype. And very recently, when the KIOPS genes were identified, another family with a different mutation in this gene, so in the C14ORF142 gene, was reported to us with the same attenuated phenotype. And so the C14 encodes a small disordered protein of unknown function, and it has exactly the same size as the yeast Gun7, but very weak sequences similarity. And thus we hypothesize that C14 could be the fifth element of the human KIOPS complex, and that was right. And in proteomics analysis and co-IP, Gun7 had the capability to interact with LAGE3, so a component of the KIOPS complex. And in addition, in lymphoblastoid cell lines obtained from patients mutated for the C14 gene or the WDR73 gene and OSGEP gene, so other genes involved in GMOs, we were able to show by Western blot that the lack of C14 leads to the decrease in the expression of other KIOPS proteins, whereas it was not the case in controls and in patients mutated with the WDR73 or the OSGEP. So this result suggested that C14, that is now named Gun7, would stabilize the KIOPS complex to modulate its function. And in parallel of this work, our group identified mutation in the last gene I told you before, that is the YRDC, that encodes for the protein YRDC. That is the first enzyme involved in the T6A biosynthesis pathway. And you remember, it's the enzyme that is able to transfer the threonine, to build the threonine carbamoyl AMP for the KIOPS complex. And we identified patients from two families that had either primary or secondary microcephaly with facial dysmorphy and hypotonia and seizures associated to hypothyroidism. And patients from the family B had a duration defect, whereas the patient in the family A had a normal MRI at 5 months. But that progressed to major cerebral and cortical atrophy by 11 months. And all patients died before 1 year. And the renal phenotype was similar with congenital nephrotic syndrome and severe diffuse mesangial sclerosis lesion and food process effacement at the electron microscopy. And the clinical phenotype was even more severe than that of the KIOPS mutated patients. What is the YRDC? The YRDC is really crucial for the T6A biosynthesis, since it's the only one that provides the threonine carbamoyl AMP to the KIOPS complex. And the assay of the pathogenicity of the YRDC mutations, to assay, we performed complementation studies in yeast, and in yeast, the mutation are unable, lead to the inability to growth in absence of the SUA5, that is the ortholog of the YRDC. And so you can see, for example, for mutation 1 and for mutation 3, that we can detect an hypomorphic allele linked to a moderate effect of the mutation, while in the mutation here, in the mutation 2, in the red box, you can detect amorphic allele with a very severe phenotype in the growth complementation study. So again, different kind of mutation linked to probable clinical heterogeneity. And as expected, the T6A level was dramatically decreased in patients with YRDC variants. So in conclusion, the take-home message for the KIOPS complex is that we identified several mutations in all the components of the T6A biosynthesis pathway. However, even if all patients presented with an early onset of the renal disease, there are some differences in the neurological phenotypes. Notably, the absence of biallelic truncating mutation in patients mutated for the 4 subunits, LH3, OSGEP, TP53, ERK, and TPRKPB, and for YRDC, which is not a subunit but associated to this complex, which would probably lead to significant lethality, and also the very severe neurological phenotype with primary microcephaly and duration and myelination defects in this kind of patients, linked to the role in the T6A biogenesis. And on the other way, less severe neurological phenotype in patients mutated for the C14, so the Gun7, the 5th element, where the brain disease seems attenuated with secondary microcephaly and the absence of duration and myelination defects, and the differences in the clinical phenotypes between the KIOPS, YRDC on one hand and the C14 on the other hand can be explained by the different role of these proteins in the T6A modification that directly affect cellular proteostasis. And YRDC is directly involved in its biosynthesis as OSGEP and the KIOPS complex, but C14 is mostly involved in the stabilization of the KIOPS complex. So the KIOPS complex could act its own role without the C14 or with an hypomorphic mutation in the C14. So this data clearly demonstrated the crucial role of the T6A modification in the pathogenesis of GMOS. Another tRNA biosynthesis pathway involved in GMOS is that of the M7G modifications, and I'm sorry, yes, the M7G modifications, and this modification consists in the methylation of the guanine at the position 46 on the tRNA, and it's performed by the holoenzyme of 2 subunits that are the WDR4 and the METTL1, that is a catalytic subunit. And so the M7 modification is a non-essential tRNA modification, contrary to the T6A modification, and so it has been shown to be involved in mRNA export and splicing and translation. And so WDR mutations have been initially described in microcephalic primordial dwarfism, but the group of Frieda Hildebrandt also described the homozygous splice site mutation in a GMOS family, and this fits very well with what has been previously shown with KIOPS or WDR73 mutation, since the WDR and or METTL1KO mouse, knockout mouse, in mouse, induce a defect in translation of cell cycle genes and in proliferation in neural lineage and differentiation of cells. And just 2 last slides on mutation in gene encoding proteins that are not directly involved in RNA metabolism. Indeed, we can find mutation in genes encoding the nucleoporins, for example, that form the nuclear pore complex, and they have been identified in several cases of GMOS. And the nuclear pore complexes are involved in nucleocytoplasmic transport in both directions, from the nuclear to the cytosol and from the cytosol to the nuclear, and also in cell division and chromatin organization and gene regulation. And actually, mutation in several NUP genes were known in isolated sRNAs. And in these 2 genes, the NUP133 and the NUP107, we found mutation in GMOS genes in childhood, in patients with childhood FSGS and primary microcephaly. And analysis in cells and animal models confirmed the implication of the detected mutations, showing decreased interaction between the 2 proteins or decreased expression of both of them, reduction in the nuclear pore complex density. And in the zebrafish model, in a morphont zebrafish model, it was observed a decrease of midbrain width and axonal number, and underdeveloped. Wow, sorry.
It's okay, we can see your slide.
Yeah. Okay, seems to work. And underdeveloped glomeruli and food process effacement in patients. And the last gene that have been described in the GMOs, in the PRDM15 gene, is really a new player in the field. And PRDM15 is a zinc finger protein known to regulate the notch pathway, and it's ubiquitously expressed with its higher expression found mainly in the lymphoblastoid cell lines, and knockdown in podocyte cell lines linked to the dysregulation of genes involved in development and cell proliferation and differentiation, such as here the WT1 or JAG1 or PAX2 genes. And again, the group of Friede Hildebrandt has identified 3 homozygous missense variants in the PRDM15 genes, and one located at the zinc finger, leading to GMOs in the zinc finger domains, leading to GMOs with specific phenotype with polydactyly and microchoria, colobome, atrial septal defects. And this variant was enabled to promote transcription in a luciferase assay. And they identified 2 another variants in another domain of the protein that lead to isolated steroid-resistant nephrotic syndrome. And the localization of the mutation in 2 different domains of the protein might explain the variable phenotype in both type of patients. So to summarize, we have talked about many things today, and we showed that the GMOs patients present mainly with mutations in the genes encoding KIOPS complex and WDR73 protein involved in RNA metabolism. And I think that you are convinced about the involvement of the RNA metabolism, and in particular the transfer RNA modification in the GMOs pathophysiology. Mutation in the new genes as well as in PRDM15 show possible alteration in GMOs, but also in other processes like with other processes, cellular mechanism like nucleocytoplasmic transport and transcriptional regulation. And neurons and podocytes are both post-mitotic non-dividing cells, are really sensitive to translation process alteration, which lead to either decreased proliferation and increased apoptosis. We have some gene mutation that might lead to specific phenotypes such as GMOs, but also isolated sRNAs, and we are not exclusive between these 2 kinds of phenotypes. It's really difficult to have animal models for this type of phenotypes, and perhaps that studies on cerebral and brain organoids and various cell types derived from IPS may help to better characterize the molecular defects in GMOs. I thank you for your attention, and of course I thank Professor Corinne Antignac and Geraldine Mollet for the huge work, and all people in the lab that did their different work on this topic, and collaborators, and of course all the clinicians for the involvement and patients' data.
Okay, so I think you announced the new webinar. So you can show that slide, Guillaume, so that people can see which will be our next webinars. And thank you so much for your excellent lecture. It's really amazing that you could summarize so much research data and so much information in such a short time frame. So really, really amazing. So please ask questions. I'm just opening my question panel. While people are thinking, I was really amazed by the amount of data, and I was wondering, because some people listening are clinicians, do you give some tips, in how people can, because the phenotype is so different, how people can suspect mutation in one of these genes? In patients, of course, you have neural anomalies and you have kidney disease, but it's largely variable. So are there specific tips that you would say that you have to think about Galloway Mowat? And please recommend.
As we showed, there is no really specific features of in GMOs, and we can find some a large panel of phenotype and clinical heterogeneity in patients. But I think that the main phenotype is really the cortical atrophy and cerebellar atrophy linked to proteinuria or nephrotic range proteinuria that really should think about the GMOs. And we have to note that in the lab, for example, for some patients with this kind of clinical findings, we have not found any mutations in the GMOs gene. So perhaps that there are again another genes that we will find in the next years. And for sure, there are two genes that are OSGEP and WDR73 that are not frequent, but in the field of rare disease, they are the most prevalent genes. And for all other genes, we detected only one family for each. And so it's really possible to detect again new genes in very isolated families.
Right. So, and another question was, because not all patients apparently have a kidney phenotype. So I think that there are some patients within neurology clinics with mutations of one of these genes that are undiagnosed, or some of these families are coming from neurologists and not from nephrologists.
Yeah, sure. I think we will detect these kind of patients soon, because I don't know in Europe, but in France, and I think it's quite the same in all the genetic departments, all patients with intellectual disability or some neurological defects very quickly go to the genome analysis, whole genome analysis. And so I think that even if the GMOs gene are not in panels, for example, for neurological defects, we will very soon know if some patients with isolated neurological defects have some mutations in the GMOs genes without renal defects. But at this time, we don't have a lot of patients with isolated neurological defects, and so it's possible that we miss some.
Yeah, thank you. I have another question from Dr. Vallet about the heterozygotes. Do heterozygotes have minimal symptoms, or are they completely asymptomatic?
As far as we know, they are completely asymptomatic.
Okay. And we have also information about this type of patients in different populations. Are all patients that you have are mostly Caucasian, or you have information in Asian patients or other races?
It's difficult to know really if we have some species, some ethnic particularities, but we detected some mutation in some populations, some specific populations. For example, the mutation in the C14 that have been detected in Algerian population. And of course, as it's a very severe recessive disease, the the disease is mostly found in consanguineous families.
Right. Yeah, thank you. In terms of the pathogenesis, I was curious, but it's a very simple question. So you said that, like in general terms, these genes are involved in the cell cycle and proliferation of podocytes and neurons, but these are really highly differentiated cells. So I wonder why these specific cells are so much dependent on cell cycle or proliferation. I would say more dividing cells should be more affected, but here it's a kind of vice versa.
Yeah, I agree. I don't know why they are so dependent to cell cycle, and perhaps it's, at least in the GMOs, I think that the main effect is not the cell cycle and the proliferation, but the ER stress and the UPR activation, and so the cell stress leading to apoptosis and to cell degeneration. But for example, for WDR73, all the work by Frances Tilley really suggests that these proteins have the capability to maintain at least the podocytes and the neuron in a differentiated state. And if you knock down this protein, you will have a regulation of other protein involved in the re-entry in the cell cycle, and so the loss of the specificity of these post-mitotic differentiated cells.
Yeah. Okay, I see. And maybe something I can imagine, these are very complex genetic defects, but also again a very naive question about the possibilities of the treatment there, because they miss a specific modification of the tRNAs. So can you imagine that you can somehow supplement this TCA biosynthesis, somehow push it by kinds of small molecules, or is something really other science fiction?
I hope that with a kind of RNA therapy we could, but that's difficult because it's a very early, most of the time it's a very early disease, and I don't know if we can reverse a phenotype in a damaged cell.
Yeah. Okay. So I just checking the panel of the question panel. I don't have other questions. So I would like really to thank you for this excellent webinar, and the audience for being here, and please follow our webinars during the next week. So thank you so much, Guillaume. Thank you. Bye-bye.
Thank you, Elena. Bye.
Thank you. Bye.
Bye.